Coming over.
I would like to thank the organizers for inviting me for this talk. So actually one of the things that you need to consider in cryopreservation is to keep the mechanical properties of a tissue. And I'm going to talk about a little bit about this mechanobiology, but also in the next half of my talk, I'm going to talk about emergence of these microfluidic technologies. So this mechanobiology field has been like on this peak in 2010 when the scientists discovered these two transmembrane protein PSO1, and simply they are responsible for how we sense forces. And this has been highlighted in Nobel Prize in 2021, but this is only the first question that they have discovered. How do we sense forces? There are lots of other unknowns that has been emerged since then, like how the cells act to these forces, how the mechanotransduction signals by the cells are activated to translate these forces to biochemical signaling, and simply what is the role of mechanical properties of single cells, tissues, but also molecular structure in these mechanotransduction pathways, and how do cells basically generate forces, and how they interact. I'm going to give you a few examples, like one of the basic fundamental concepts in the life is cell division. If you have perturbation in cell division, you're going to have cancer, but also you're going to have accelerated aging. But if you look at this concept here, this happening through this fine-tuned oscillation of this microtubule that are connected to these chromosomes. And it's been shown that this oscillation is very, very fine-tuned, and if it's perturbed, you're going to have failure in cell division. And the question here is like, how do the genes control this phenomena? And what is the role of mechanical properties of these structures, but also the cytoplasm of the cell in the cell division? So we recently discovered that like, if you change the viscosity of the cytoplasm, you're going to have failure in cell division, you're going to have delayed in cell division, make it more viscous like honey compared to water. It's going to get failure in the chromative velocity is going to be basically slowed down. And then you're going to have failure in cell division. You're going to have less immune cells in certain type of cancers. Another example that is fundamental is in aging, but also in cancer, lots of diseases, is this migration. So the migration is obvious. For example, for immune response, the immune cell is going to be following the bacteria, for example, in this case. But how they follow this? They sense the chemical signal from the bacteria, and inside the cells you have this complex cytoskeletal element gonna reorganize themselves to follow. And any failure in this mechanism gonna lead to failure of this following, gonna have immune perturbed or changed or altered immune response. And so the question here is, what of mechanical properties of the cell, because this is like a machine. If the machine is soft or stiff, it cannot generate the same force, it cannot move the same way. And then how the cell basically do these processes, and what's the role of mechanical properties of cell itself, but also the cell is not moving in a simple environment, sometimes moving fluid, sometimes moving inside the 3D extracellular matrix. And the last example I would like to mention to highlight the role of mechanobiology from fundamental biology, but also aging, is this: when you put cells inside different mechanical environment, they're going to respond differently to the environment. So in this example, for example, around 15 years ago, 20 years ago now, the scientists put stem cells on top of different metrics with different stiffness, and simply can differentiate the stem cells into different cell types by putting them inside different mechanical environment. So it's not all about genes and biological pathways and chemical interactions. It's also about mechanical environments that are regulating the cell behavior. And this is the general topic of mechanobiology. And then in the context of aging, I want to like define this mechanical hallmark of aging. There's a lot of attention in this scale, in the molecular scale, in terms of, we know that there are lots of DNA damage, chromatin remodeling, and protein reorganizations, and there's lots of large scale. So we know, like, when you have diseases, like you have problems in your bone, muscles, teeth, vasculature, heart problems, but there's been less of attention to this intermediate scale, which is like from few nanometer to few micrometer scale, which is the cellular interaction and mechanotransduction. And in the aging, as explained in the previous slide, you have a change in the extracellular metrics, you have a change in your tissue mechanical integrity, and if you look at these pathways, you're going to have perturbed pathways. There is some recent review on the topic of mechanobiological changes in aging. Today I'm going to focus on one aspect, which is only stiffness. Briefly I'm going to talk, but the problem with that scale is you need to have tools to measure force and stiffness at the scale of micrometer. So it's not easy to measure, for example, cellular forces. We've been developing this technology called traction force microscopy on the bottom that you can measure nanometer, picometer cellular force generation. But also, I've been over the last 10 years on this system called atomic force microscopy, which enables us to measure mechanical properties of single cell, but also you can run very high resolution imaging depending on the tip that you use. So here is the concept. It's very simple. You turn the deformation of a cantilever into change in laser light paths. So you can measure small nanometer and sub-nanometer deformation using this technology. And then using different tips, sharp tips or spherical tips, you can measure nanometer scale structure, mechanical properties, or in this case, you can indent a single cell and measure how soft or stiff it is. You can apply piconewton, nanonewton forces, and you can measure nanomicrometer deformations. So we applied this technology recently on a tissue scale level and measure particularly the mechanical properties of central nervous system. And why mechanical properties of central nervous system is because it's one of the softest tissues in the body. And again, coming back to cryopreservation, when you freeze this brain, you're going to have lots of changes in mechanical, irreversible changes in mechanical properties. So we developed this technology that you can run on top of a tissue and measure the mechanical properties of tissue at cellular resolution. And on this slide, you can see the spinal cord of a mice, coronal and transversal cross-section, and these are the mechanical maps. It's quite soft, 200 Pascal, 300 Pascal, which is like extremely soft. The brain is extremely soft. And the interesting thing is, you have to measure these things live in a certain condition. So for example, if you freeze the tissue or use a PFA to fix the tissue, you're going to lose all of this structural heterogeneity in terms of mechanical properties. But also we can correlate this mechanical properties nicely with cellular structures. As you can see in the white matter and gray matters, you're going to have inhomogeneity in the mechanical properties. And recently we published a paper showing that glial skulls, which is happening after the injury to central nervous system, is a softer tissue than before scarring. And this is kind of counterintuitive because normally when you have injury into your body, you expect it to be stiffer tissue because of fibrosis. But in the brain is the other way around. So moving on from CNS injury and glial skull, we wanted to know what's happening to the mechanical properties of brain during aging. We know during development, as your brain get more myelinated, the stiffness of the brain gonna get increased. But on the other hand, when you get Alzheimer's disease and accelerated aging, the mechanical properties of the brain decreases. The problem is the only technology that can measure mechanical properties of large-scale tissue such as brain is MRI, but the resolution is only millimeter, which is far from the resolution that we would like to achieve for cell mechanics and cell mechanical properties. So we use AFM technology, a mechanical map on this mice model of Alzheimer's disease. So we look at the mechanical map of only hippocampus, which is the major part of brain that is responsible for memory formation and cognition, and there's going to be lots of changing happening in this tissue in terms of and the development of different problems during aging. Particularly, we saw that like as we age, reaching the adulthood, kind of like from the adulthood to aging, we're going to see specific regions are getting region are getting for example, stiffer. The accelerated aging, the stiffness drops. This region is, for example, this dentate gyrus is the part that is responsible for formation of new neurons, neurogenesis. And we saw, so as we get to the adulthood, the stiffness increases, but as we get Alzheimer's disease, the stiffness is dropping. Interestingly, we could correlate this stiffening and softening with the expression of GFAP, which is again something that is happening with aging and also Alzheimer's disease. We could correlate the molecular, the cellular mechanical properties with the biological mechanical properties. So this was the first part of my talk. It was about tissue stiffness, cell stiffness, and how it can be correlated and why is it important. In the second part of my talk, I would like to talk about this microfluidic technology. This is something that has been well established now in the field of, for example, cancer, but has been less mentioned in the field of aging. So I would like to use this opportunity to introduce. So there's lots of things that you can do with these small scale channels that can hold small volume of fluid, that you can do lots of analysis at a single cell level, but also the most important thing is you can have small volume of samples for analysis. And we know that single cell analysis is something super important in biology, and they're all happening through these microfluidic channels. What I would like to give an example today is this organs on a chip technologies, which is part of microfluidic technology, but it's a little bit larger scale. We are not going down to 10, 20 micron. We are going to talk about a little bit bigger, 100 to 200 to 1,000 micrometer channels. So we can put cells inside these fluidic channels in 3D environment embedded inside extracellular matrix. And you can have a super control over their structure and their intensity, and you can apply, for example, flow, pressure gradient, chemical gradient, and different other things to this system. I'm going to give you an example later, a clear example of how we use this technology, but some of the main advantage of this technology is that you can put the cells in 3D culture system, but you can run high resolution imaging. This is the most important thing, because when you go to 3D, it's not gonna be easy to, for example, in either 3D cultures or also in animal or human to do live imaging, because the light cannot penetrate more than 200, 300 micron. And we know for high resolution imaging, the only technology that is gonna be very useful to do the thing, to investigate biological phenomena is light microscopy. So these chambers allow us to put cells inside 100 micron, 200 micron channels, and we can apply flow, we can apply pressure gradient, and all kind of intervention while we are doing live high resolution imaging, and can be a bridging gap between these simple 2D culture systems and animal models. And we know these 2D simple culture systems are so simplistic because the cells are not sitting inside a stiff Petri dishes in the real body. They are inside a 3D environment, experiencing a complicated environment. So I'm going to give you one example for the last part of my talk of these organs on a chip technologies. I've been getting familiar with technology when I was doing my postdoc at MIT in Roger Cam group. They were using these microfluidic chips. They call it 7-channel device. They put endothelial cells embedded in fibrin gel in the middle channel, and then they put fibroblasts on the side channels, and there's a paracrine signaling happening after 4 days. This vasculature, 3D spontaneously formed vasculature is made up, and then they use this system to perfuse cancer cells from side channels and look at the extravasation of cancer cells outside these vessels. So they use this small chip. The chip is quite small. The height of the chip is around 100 micron. The width of the channel is 1,000 micron. And so they were using this chip to study the process of vascularization, but also to look at cancer metastasis, processes related to cancer metastasis. So my question as an engineer or bioengineer at the time was, can we make this bigger, because this was so small? Can we make this engineered tissue bigger? Because micrometer, everything in your body is millimeter, centimeter. Your vascular system of micrometer is quite millimeter. So I tried to increase the system. So we increase the channel sizes now here by 3 millimeter, and we realized that there is no way that we can form a nice, perfusible, interconnected vasculature. And we first thought that it's a problem of diffusion, because the cells that are on the side are having maybe lack of signaling to have signals communicated to this endothelial cells in the middle to form vasculature. But then if you look at perfusion of dextran in this setup, it's going to take only a few minutes, 10 to 16 minutes for all the secretion factors to get perfused everywhere. So that was not a problem. So what we thought is maybe instead of putting cells to signal in a paracrine way on the side, we can mix the cells and look at what's happening. So amazingly, when it mixes the cells, fibroblasts and endothelial cells, in a large-scale devices, we can make this amazing vascularized system that is fully perfusible, and it has certain features that you cannot achieve when you put the cells in the paracrine system. So we try to characterize this system and seeing what's happening then when you put cells either in the endothelial cells in the monoculture system or in the paracrine system, or when we put them to interact physically together, what's happening that you're gonna have in this system only a very, very robust phenotype of vasculature. And during this analysis, we realized that when we are removing this vascularized tissue, these are now large-scale vascular, I would call it large-scale vascularized tissue, but there are no way still comparable to your organs tissue. When we were removing these tissues, for this vascularized system, we could stretch it up to 6-7 times without any rupture, and we could stretch it like back and forward very robustly. And this is like something that was not possible with if you have only hydrogel, or if you have only hydrogel that you put bad vasculature in, that there's a bad vasculature form inside. So only this very, very robust vasculature that had specific mechanical specific phenotype had a very, very robust mechanical feature. And this is what happened in your body. Your body is extremely robust. Your vasculature, your organs, your heart, you're all vascularized. Your brain is vascularized tissue. They are not going to be failing easily by small deformations. So we tried to characterize this system better with atomic force microscopy, and we found out that the emergence of this highly stiffness mechanical phenotype is only happening when you put the fibroblasts and endothelial cells together to interact together inside the 3D environment. If you put these cells individually or in any other combination, you are not going to get this amazing phenotype, this stretchable phenotype, and this highly stiffness phenotype. So in the system that I showed, I think I didn't clearly show there are fibroblasts and there are endothelial cells. They are interacting in a Juxtacrine format. The system has very, very low permeability, similar to the tissues that you have in your body, vascularized tissue that you have in your body, but also it has a very, very strong mechanical phenotype. And in terms of network structure and perfusibility, it has high degree of similarity to real tissue. So in recent years, we established this company 199 Bio, and then we try to see if we can use this technology to have a model of aging. And simply what we did is we took fibroblasts from old individuals and put it in the system, and what we saw is you can see there's a failure of vascularization. So simply changing the fibroblasts from young individuals, the skin of young individuals to old individuals, 70 years old. You can see the vasculature form. The vasculature is forming, but it's not perfusible. It's not fully interconnected. So the idea here is what kind of intervention we can do on this fibroblast to rescue their phenotype. And the idea here is very important because we are having a functional feature. It's not about application of different drugs or intervention single cells and measuring their clock or epigenetics or methylation analysis. We have a real functional feature. And what we discovered is if we do partial reprogramming either through lentiviral transduction or mRNA treatment, if we do transient partial reprogramming on this fibroblasts, we can completely rescue the phenotype of aged vasculature. And this is kind of like a very, very important system because we have a model of aging, and the idea is now how can we nail down the treatment specifically for vasculature. And why only vasculature? Because vasculature is part of all the organs in your body. And I was talking with some aging scientists last week from US, Tamir, and he was apparently vascular endothelial cells are one of the highest senescent cells in the body. So if we can rescue their senescence, kind of to make them younger, we can have a better vasculature. Automatically, you may solve lots of problems in the aging problem. So what we are doing now next is the same model that we have for vasculature. We have the model of blood-brain barrier, which is representing your brains. Instead of fibroblasts, we have other brain cells like astrocytes, pericytes, and then we are trying to see if we can rejuvenate the brain vasculature, but also we are trying to see what we can see here in this mini organs on the chip we can see in the animals. And in addition to vascular system, we have a myelination model on a chip that we can test to see if the similar intervention can also result in better myelination of myelinating cells. So if you are more interested about the work of the 199 Bio, there's going to be another talk from our CEO, Boris. I think it's going to be on Thursday, 9:30 to 9:50. If you are interested about the company work, please join that talk. Otherwise, I'm happy to take any question.
Thank you very much. We've got time for 2 questions.
Here. Very
okay.
nice talk.
Thank you.
What is the signal from fibroblasts sending to the epithelial to interact?
So there's lots of, as I mentioned, there's lots of chemicals, paracrine signals, lots of molecules, but in this case, there's the mechanical signals, which means these fibroblasts are pulling upon the extracellular matrix, and these forces are sensing. The paper that we published, we are showing that these forces that are exerted by fibroblasts are transmitted to endothelial cells. So it's basically real, actual forces that are transmitted. So when you have cells in the body, they're either going to secreting signals or they're going to be mechanical forces. And the thing that is missing in biology, specifically biology of aging, is mechanical signals. That I would wanted to highlight.
So the reprogramming is actually reprogramming the force.
The reprogramming is reprogramming the fibroblasts, which then reprogramming the fibroblasts going to reprogram the secretion factors, but also the forces.
So just in line with this one, so my question would be, is it the failure of forming a proper exo matrix with the old fibroblast? Or is it, can you test this? Can you decellularize it and then put...
So in the original paper that I showed you, we've done all of this process. We decellularize the matrix. We look at the matrix composition. We could look at the matrix reorganization. In this experiment, these are just the results that we get a few months ago. We are trying to see what's happening, but what's happening is secretion factors, but also ECM remodeling is going to be failed. What other things that we are doing now is to do traction force microscopy on this fibroblast to see if these fibroblasts going to lose their force generation or they're going to increase the force generation. It's not obvious. Some communities, some experiments shows when the cells go to senescence or old phenotype, they have increased forces, but we don't think it's going to be increasing. It's decreased forces.
Fantastic. Thank you, Emad.